Okay. Can you hear me in the back? Okay, good. So this is the third lecture of a series of quantitative bacterial physiology, complementary to mass cost lectures, focusing on the cell cycle and the coordination issues. And the last two lectures, we talked about several different things. But by the way, just one diversion. I was supposed to meet several students over dinner yesterday, and I didn't show up. So I apologize for that. And then right after my lecture, I went to my bed, and then I just crashed, and I couldn't get up. So, but I'm going to be around the whole week next week. So please do come and find me, and then I'll be very happy to talk to you, okay? And about anything. Okay, so my apologies. Coming back to the recap, we talked about several different things, but the underlying theme was that it's about coordination issues, and then the consistency or self-consistency check between different logical framework. Okay. Our starting point is always the experimental data. As someone trained in physics, I like theory, but I've entered biology in a very hard way. And then in the end, after all these years, one thing, if there's one thing I've learned, that is biology is an experimental science. Okay? Theory is very important, as Professor Pippel said. In fact, I'm looking for a theory postdoc. But even then, I think there should be a very close interactions between the experimental the scientists and those who are interested in theoretical aspects of biological systems. Okay, so even for those theoreticians, I strongly recommend you try to get your hands dirty, at least at some point in your life. All right. So our first starting experimental data set was from the 1958, the growth law by the people in the Copenhagen School of Bacterial Physiology. Very simple and beautiful rule that the size of the cells, on average, are correlated in an exponential way with respect to the growth, average growth rate of the cells in steady-state growth conditions. Okay? So when you didn't know anything, we simply expressed this as a one exponential functions like this. The same year, 1958, there's a second paper in the same journal, which was about the transition between two steady states. So we define point A here, and then the question is, if you change your growth environment very suddenly, how do you go from cells go from point A to point B at this point? Okay. And then we played with some math, defining the size as a ratio between the optical density, OD, and the number of cells. Okay? So this is the actual data from the second 1958 paper. Okay? You remember these curves? So the x-axis is the lab time, the actual time in minutes, and y-axis is the axis for various parameters. This particular line was the optical density. It is about how fast the biomolecules synthesize and multiply in a tube. And as soon as you do the shift up, the rate of synthesis increases immediately, almost immediately, to the new steady state level. Okay? That is the OD. Then, if you follow the amounts of DNA, there's some lag here, and then after some lag, the synthesis rate reaches a new steady state value. We also talked about how the number of cells increases and their synthesis rate. Again, there's a long persistence, and then it shifts up. We talked about this gap here from the moment the shift up was done and the moment where this new growth rate kicks in. Okay? So by doing some calculations and trying to enforce consistency between the growth law here and these observations, that you have essentially two exponential lines here, we deduce that this delay is exactly this gamma itself. And up to still that point, we don't exactly know what this gamma is about, okay? It is just some constant which has a unit of time. Okay? And many years later, Helmstadter made a lot of effort to obtain the synchronized cell populations, and he developed the celebrated baby cell machines. We talked about it yesterday. Up to this point was okay because this is like a cartoon. So you have cells of all age groups in the steady state populations. You use a filter, so you filter through this cell culture, and the cells are stuck on this filter. You flip it, and you elute with the fresh medium, and then the cells start to continue to grow, and then you can collect the cells, number of cells in a specific stage between birth and division in a synchronized way. Okay? So this was the Helmstadter's vision. Then he made, after many years of experiments, he made it work. And then the obtained populations really reflected the theoretical one that one would expect, which has exponential decay. Okay? And he teamed up with Cooper and came up with the so-called, well, I don't know whether anybody actually calls this way, but from what I always call this as a two-timers model. Two timers intrinsic to the cells running in parallel, but independently. The first timer, which is about tau, which measures the generation time, triggers the initiation, new round of initiations of replication at every tau minutes. Okay? But you also have to divide at some point, and that is done triggered by the second timer, okay, which is a C plus D. C was the duration for replication, C maybe for copy, and D is the G2 phase in eukaryotes. It's a duration between termination of replications and cell division. Okay? And if you really think about it, you'll realize immediately this is extremely, extremely fragile model. Okay? Just like any timer-based models, there's no really stable points. Okay? So once the cells make a little bit of mistake, there's a little bit of noise gets introduced, then everything breaks down. Okay? So perhaps I should come back to that at the end of my lecture as one of the, from my point of view, really great open questions. We really don't actually have a good understanding yet. Okay, and then the shift up stories we talked about. Yesterday we talked about how one can explain this kind of radioactivity signals between births and divisions, okay, using the Helmstadter-Cooper model. And I apologize, yesterday this was a bit too fast, but as I said, this multifold replication is inherently complex processes, okay? So it just takes some time to get used to think like Helmstadter-Cooper did in the 1960s. But, you know, if you spend some time, you'll get used to it, okay? So I will distribute the lecture notes as soon as I go back to the US, and then there are some calculations you can actually play with, and then you'll also see what kinds of holes, logical gaps are in those arguments. Yes?
So it doesn't go above 6, namely no more than 3. It doesn't go above 6 and no more than 3 forks at a time. Does this suggest that you never nest it even further? And if that's the case, does this mean that there is another rate-limiting step that you would reach, such as, I don't know, production of ribosomes? So the DNA is no longer rate-limiting.
So the question is about what really sets this upper bound, for example. And the answer is, I don't know. Okay? But you know, if you talk to people who study E. coli, they always tell you, oh, E. coli can grow as fast and divide as frequently as every 20 minutes. as if that is some kind of a really the absolute the fastest frequency for division one can achieve. And then, to tell you the truth, if you use so-called soyton, which is a kind of vegetarian soup for E. coli, you can actually achieve something like 17 minutes of generation times. Okay? Studying the actual cell cycle for the 17-minute generation time is a little bit trickier somehow. Okay? But empirically, we know that we can actually go faster than that. Okay? So, if you're a theorist, you can actually try to rationalize what cell set the 20 is. For example, here, the Mascott's work in the 2010 also actually suggests what may set the upper boundary. Okay? So based on the resources available in this proteome, based on this linear relationship between the growth rate and the ribosomes contents, and etc. So we can actually rationalize, all this, but I think the real mechanistic answer, as far as I'm concerned, I don't have the answer for you. Okay? So this is one particular data set, and then in different growth conditions, these curves will look different. Yes? Okay, I can't hear you very well, but if I understand correctly, how did they measure the protein concentrations? So to simplify your question, what did they exactly measure in the 1958? And the answer is here. So the first thing they measure, from the top, they measure the RNA content, then DNA content, and then number of cells by counting the colony. But proteins are not there. I don't know why they didn't actually measure the protein concentrations, although, as you must remember from Mascott's lectures, people really did measure protein, the level at different concentrations by Neinhardt, all these people. Okay? So in this particular study, in this graph, that parameter is not there. But why are you interested in that? Why are you interested in the protein content? Are you interested in the concentrations of proteins, or are you interested in the absolute amounts of proteins? So are you interested in the concentration of proteins, or are you interested in the absolute amounts of proteins? Are you interested in absolute amount? Okay. For that, for now, let's assume that the optical density gives you some hint about the amounts of proteins in the culture. Okay. Any other questions? After these recapitulations, essentially, I'm going to give a research talk for 1 hour. Okay, and I'll invite you more questions. Yes. This is for the reference. Okay, just to guide the eye, so that to tell you that this slope is the slope expected for the new steady state after shift up. Okay. Any other questions?
Which one of these should it follow?
I will repeat the question. So yeah.
Okay. So suppose if we do the analysis for protein, what should it follow? Should it follow the RNA? Because according to Mascott's lectures, RNA, then it's proportional to rRNA, and then it's proportional to the somehow it's proportional to the protein.
Right. So the question is, if we did have the protein levels as well, what should we expect? Okay. Do you have any suggestions? Why is that? Why should we follow RNA? Okay. The suggestion here is that the protein amounts should probably follow the RNA amounts. That sounds reasonable, although we should also consider some time delay for the protein synthesis. Do you have any suggestions? Yes. the rate limiting steps are all very important. Does it come from the speed, or does it come from the availability of those machinery, etc.? Yeah. They're all coming to the picture. Okay. Any other questions? Yes. Yeah. I should have probably masked that one, because this is 1958. And that was when the cell biology was very in the early stage. So they probably thought that even bacteria have a nuclei. Okay. So I don't remember exactly what they meant by that. I think they actually meant again, essentially probably kind of chromosome-like object. Okay. But for now, let's not pay attention to that. Yes.
So if I see this correctly, there's a delay in there's a shift in the slope because of the initiation, right? Which happens 10 minutes post shift up, and then 70 minutes post shift up. There's the delay in division, right? That would mean that the difference between the two delays is about 60 minutes, which is what C plus D is, right?
Right. Except the C plus D. So in the classical literature with a B bar strain of E. coli, we say C equals 40 minutes, D equals 20 minutes, thus C plus D equals 60 minutes. But that really depends on the strain. Okay, and this is not even E. coli. This was a Salmonella, cousin of E. coli. So I don't remember what exactly the C plus D was.
Yeah, but now that you're saying this, you think it should be about 70 minutes?
Yes. So if we take this seriously, then I'd expect this to be C plus D would be 70 minutes, exactly where that number is.
Okay, thanks.
Without considering all the first of the perturbations I mentioned in my first lecture. Yes. Right. Yeah. So yeah, I realize even more. So his question had two parts. The first part was the clarifications about slow growing conditions and fast growing conditions, and whether we talked about we defined BCD period, and the question was whether the B period is actually some fundamental cell cycle stage, because when slow growing cells B period is there, fast growing cells we only talk about C and D, and the short answer is that as far as the bacterial physiology and cell cycle is concerned, B period is not a fundamental cell cycle stage. What is important is a C, the replication, and then the gap between termination and division, D period. Okay? So that's the first part, clarification. The second part is about the same question: what exactly should this number, and should there be any specific relationship between the time point, this kink here, and then kink here? Okay, and short answer, without knowing anything, would be probably, because all these lines can be in principle calculated by assuming Helmstedt-Cooper model. So I'm sure you can just calculate it. I don't remember what the answer was. I strongly encourage you to play with that. Okay? But even without thinking about the Helmstedt-Cooper, I emphasize that this delay from shift up should be simply gamma as far as the average is concerned. Okay? That's just a mathematical. We can actually rationalize it too, but probably I shouldn't do it right now. I pressed something I shouldn't have. Okay, good. Sorry. I fixed the problem. Created a problem and I fixed the problem. So this was the shift up that I just couldn't reproduce this very well in the whiteboard, but you already get some idea. So this is the A state and this is the B state, and the semi-log scale cell grows and divides and grows and divides, and this is a tau A. Okay. At some point, you do the shift up, okay, and then the growth of the cell basically increases immediately after shift up like this, divides in the middle, increases, and etc. The key point here is that no matter what the growth condition is, there's always a C plus D gap or separation between one round of initiations and cell division. Okay, so this is really the hallmark of a Herrstadt-Cooper model. Nobody knows where that comes from, but if you accept it, you can explain all the shift up data. Yes, question there. Right. Right, in this particular case, okay, but remember, this is for realization of a single equalized cells. And then in a steady state growth condition, the cells can be in a different state. At this particular point can move. No. So average. So can you repeat that? Uh-huh. Yeah. Where is TS? Right. So this is correct. So this is the lab time, right? This is the lab time where the transition took place. Yeah. But I think that you can get we can talk about all the details. But I think what is the most important thing here is that they're really conceptually before shift up. There are always a separate the time points where division took place, okay? But that division before shift up still dictates the cell division until after C plus T from that point, okay? The largest time scale one can expect, okay, as a kind of memory before shift up is really determined by the length of this arrow, which is C plus T. Okay? So that will be the last point of this, and then that will still dictate the division up to C plus T. I think this is what is most important. Okay? And you are more than welcome to do bookkeeping and then calculate it and then check my note and see whether the numbers that come out of that is consistent with this argument. Okay? And I haven't done that check, but I'm pretty sure you will get the consistent results. But correct me if I'm wrong. Okay? Yes.
The cell size at the time of initiation in the blue before shift up seems to be smaller than the cell size at the time of initiation in the green one.
I completely lost you because I missed the first three words. Can you please repeat that?
The cell size before shift up.
You're talking about these lines.
Yeah, before shift up.
Yep.
Seems to be smaller than the cell size after shift up. So look at the second green point.
Second green point, this one.
So at that point, the cell size, the size of the cell at that point seems to be larger as compared to the blue point, as compared to when the initiation started before shift up.
This point.
No, initiation, yeah, so the blue point. Slightly right. Cell size at initiation seems to be slightly larger.
Yep. because in state B, the cells are larger. And then during this transition, cell size increases gradually.
Yesterday we derived the condition where the cell size at initiation seems to be constant.
No, that's confusion. Right. So let me talk about donor key. Just hold on. Okay. This is the last part of the recap, and which is here Willy donor key's analysis of a so-called constant initiation mass. Okay, it's very important questions. So what Willy donor key is very simple. He assumed both the growth law and then Helmholtz-Cooper model, and he simply wanted to first graphically figure out what is the size of the cells at replication initiations. Okay, so let's look at this particular line, for instance. Again, x-axis is the lab time, and y-axis is the size of the cells. Okay, and semi-log scale. So what this tells you is that the generation time is 60 minutes. So cells grows, and after 60 minutes, and then you divide into 2. Okay? And he repeated this graphically for different growth conditions. This time, look at this. This is generation time 50 minutes. Okay? And cell is born at certain size, which is given by this data. Because this tells you, for given growth condition, generation time, tells you what size, average size we should expect. So the starting point is, and then you grow at this rate, okay? And then you determine what the initiation point is using Helmholtz-Cooper model, which is always C plus D minutes prior the cell division some point in the future. That's what he did. Now look at this. You go to 40 minutes, 35 minutes, 30, 25, 20, and so on. Okay? And then if you look at where the initiation takes place, in this case, you see 2 groups. First, there are group cells where the initiation are all on a single horizontal line, where the cell size is 2 times some unit. Okay? In this case, for 20 minutes and the 25 minutes generation times, the initiation points are again on the horizontal line, but the total cell size at, for these conditions are 4 times some unit. And the constancy comes from counting the number of replication origins in those growth conditions. And what this tells you here is that we should expect 4 replication origins, and we should expect 2 replication origins. So if you take the ratio between of the size and the number of origins, then you realize there are 4 origins, and then this goes to 1. And here you have a size 2, but you also have 2 origins. So size per origin becomes 1. This is the constancy we're talking about. And that number suddenly, after Donakis analysis, you see, here we didn't know. The only thing we really understood was this one, which was the growth rate. But with the shift up, and Donaki and Helmstetter, these constants start gaining some meaning. Gamma, for example, became C plus D, and S0 now is the size at initiation per replication origin, okay? Which is constant irrespective of the growth conditions. Okay? Any other questions? We briefly talked about how the cell morphology reflects the shift up as well, because before shift up, the cells are small and skinnier, and after shift up, you're bigger and fatter. The question is, what actually happens in the intermediate stage? So this is the old work by Konrad Voldering in the University of Amsterdam. And what you see here is something like this. Normal-looking cells, but with a tapered cell poles, okay? Which indicated that the growth of the cell, at least the skin, is not really homogeneous. Okay? So where near the cell pole, the turnover rate of the molecules in that region is much lower compared to the rest. Okay? So basically, the tips don't grow anymore. So this is the kind of modern version. Experimental test is unpublished results from our lab of that. So you start with the very slow growth conditions. At some point, you shift up, okay? And then cells become bigger immediately, longer and fatter, but still see at the end, the tips are very tapered. Okay? So this is direct visualizations. Okay, so what I want to talk about today is that first of all, I want to skip 30 or 40 years in time. And the simple reason is that I think Matt also talked about it. The golden era of the bacterial physiology or quantitative bacterial physiology was from the 40s to 1960s. Well, in 1970s, there was still some work, but that was also the era where molecular biology really exploded, okay? And completely wiped out the biology. And all those phenomenological view of biology somehow were considered a second-rate science, okay? And just disappeared. And only in the last decade, actually less than that, and the people from physical sciences coming back to these questions and going back to the old literature. And if you read as a physicist, you read you pick up the random paper from the 1980s about molecular biology, you try to read it, okay? And you can't read it. But if you pick up the paper from the 1940s and 50s in biology literature, and somehow you can read it. It's strange, okay? So maybe that's why people liked it, I don't know. So with the birth of systems biology, this became really one of the main topics again. So this is the one question we have is that how does the cell control its own size, okay? Or do cells actually know what size they should be, okay? And cell size is a very holistic parameter because in the sense that if you change something inside the cell, some crucial part, it will reflect it in the cell size, okay? You mess up with the DNA polymerases, for example, and then that will be reflected in the cell size. You do something bad, mutate to the topoisomerases, which has to do with the DNA topology and changing DNA topologies. But again, if you do that, cell size will change accordingly. Okay? But it will be observed to call topoisomerases a cell size control genes. Okay? So that's why if you take a traditional genetics approach, cell size is a nightmare to answer. Okay? We should take a different approaches. And if you think about these guys, you already get some idea that without knowing without having to study individual genes, we still can say something about the size. Okay. So look at this one. On the left-hand side, we have a picture of two populations of E. coli cells. Again, the bigger and fatter guys, and then narrower, skinnier, and then shorter ones. Okay. And the right-hand side is the actual data. I relabeled the axis to translate to the modern language that we are more familiar with. Physicist-friendly axis labels. So, but this is the actual data from the 1958. Okay, look at this. Each dot was a one independent experiment. So they worked really hard. Imagine all the work they had to do. Okay, without any automated equipment we have these days. But end result is beautiful. Okay? And this follows here the straight line in the semi-log scale. So the exponential function is a good description that links size and then growth rate. And the reason we physicists like this is that despite the fact this is correlations, it actually implies some predictive power that we still have in biology. What I mean by that is as follows. Okay? Imagine you pick one of these bugs and then put it into a liquid culture whose chemical composition we have no idea. Okay? But all we know is that this bug will grow in that growth medium X. Okay? So you shake it, you measure the growth curves, and you can measure the growth rate of the bug in the particular medium. So something like this. What the growth law tells us is that without having to look at the cells under the microscope, we know exactly how big they should be on average. Okay? So this is the quantitative predictive power that we are very interested in. Okay? And this is the population level from the 1950s. And the question we have is there anything we can predict at the single cell level? Okay? So this is the time-lapse images of typical E. coli cells growing in a very healthy conditions. Cell is born at this size, elongates, and then the septum is formed, and it starts invaginations, and at some point, this is ready to divide. What can it predict when we look at things like this? Okay. And this is one of my favorite pictures of Francois Jacob and Jacques Monod, and already in the a long time back. And Jacob said, the dream of every cell is to become two cells. Okay, this is a very well-known remark. But if you really think about the, what this remark really underlies, okay, it implies a lot of fundamental questions. Okay, how do they grow? How do cells control the cell cycle? How is the growth coordinate with the cell cycle? How do they respond to the environmental cues? Okay, et cetera, et cetera, et cetera. And many of which are still fundamental questions that remain unanswered. Okay? And to physicists, this kind of remark actually resonates if you really think about the real meaning. A few years ago, when I entered biology as a physicist, I still had this single molecule biophysics view, which really dominated the biophysics around that time. So that I wanted to actually build something to study biology like a biophysicist. Okay. One particular question I had in mind was that can we construct kind of artificial cell-like environment in which we trap the real chromosomes directly isolated from E. coli cells, okay, and then confine it in a physical space like this, and then close with the beads, and then physically manipulate these chromosomes using optical trap. And eventually the goal was to replicate these ex vivo chromosomes and then see whether we can create kind of a real artificial cell which will self-perpetuate, okay? Grow and divide and grow and divide. What are the conditions? Okay. And turns out that this system became really useful for addressing a lot of other questions, for example. In this particular example, we use this kind of devices first to insert the E. coli cells and then make it grow as a filament. And then you simply send the hydrodynamic flow in this microfluidic device. And what happens is that this hydrodynamic flow, the Stokes force is large enough such that you can actually morphologically deform these growing E. coli cells. Okay? And then we're able to quantify how much forces are required to change the shape of the cells and in the presence and absence of actual growth and so on. Once you establish this kind of experimental system, it becomes a routine. And in fact, anyone can learn and use it to simple experiments. Okay? And here, this particular student came from Germany when she was a high school student, and then for one summer. And then and these are the two undergrad students. And they spent something like 8 weeks, 10 weeks. And they were able to do this kind of experiments. Okay? Should have been co-author, but anyways, they were pioneered this kind of scientific questions. The reason this device eventually known as the mother machine, as opposed to the baby machine, is that once you insert the cells to growth channels like this, okay, and if you think about how the cells are aligned in this device, and then if you think about their growth and division patterns, you realize the cells at the end, whenever they divide, they inherit the same pole from the previous generations. So if you construct a linear tree, what you end up following is that this leftmost line, which is this mother cell, quote unquote. Okay? So because we can retain and observe the fate of this mother cell for a very long time, this was known as became known as the mother machine. Okay? This device worked pretty well to study growth of E. coli from the very beginning. And this was almost our experiment number one. Okay, I don't know why it works so well, because in general, in experiments, nothing really works. Okay, so we are very lucky. And here, we are watching the E. coli cells expressing yellow fluorescent proteins constitutively. Okay? This is a very high throughput system. And this is one of the oldest movies we made. Okay? It's more than 5 years old. But whenever we watch it, we discover something new here. And I put some background music because there's something musical about this. Okay? The way this experiment works is that our device has like tens of thousands of channels. And then you choose one field of view and take one picture. And using automated stage, you go to the next field of view and take another picture immediately. Okay? You do as fast as you can. And in this case, our constraint was that we wanted to take pictures, as many pictures as possible within 1 minute time interval. Okay? So in this case, it's 12 fields of view. And you go back after 1 minute. And then you repeat this. And in those days, typical experiments lasted for 3 days to, say, 1 week. So that initially, you start with every channel filled with cells. But eventually, after 3 or to 1 week of experiments, all cells eventually die. Okay. Any questions? Always the same question, number one question. Yeah, of course. We are being very honest here. And this is to show you that the device is not perfect. Okay? So there's some gap. Sometimes the binding between the PDMS, the material for microfluidics, and the glass is not perfect, and they're creating some small gaps. And E. coli loves small space. They grow into the space, and when they do that, for whatever reasons, they seem very stressed. And then they express, this yellow fluorescent proteins like crazy. Any other questions? So there's another important popular questions. So initially, this device has a kind of sponge effect. The liquids just kind of wetting. So liquids follows the line of growth channels very gently, and it takes like half an hour for the cells to follow the flow line and then eventually fill the channels. But we got very lazy, and what we do now these days is that we simply centrifuge the whole device. Okay? And within 1 minute, basically, the loading rate is 100%. Okay? So this is, our throughput is at least an order of magnitude higher now. So our goal is, when we try to publish something, our goal is 1 million cells, which is at least 3 orders of magnitude larger than what people typically publish. We have a lot of quantitative power and data. Any other questions? No, we don't glue anything. Simply, this is a geometrical confinement, and the way the channel and then is set up, and then cell shape is like, so that the mother cells is just physically confined, not attached to the walls. But to do that, of course, we had to engineer the cells a little bit so that they'll make them non-motile. You have to basically shut down the motor. This one. It seems everybody knows but me. This frame first. This frame. Yes. This one. This guy. This one. As far as I see, as far as I see in our experiment, there are no persisters. Okay? It's just a dead cells. In fact, we do a lot more experiments, and in fact, we have very good ideas mechanistically what kills the cells, what makes the cells age, and etc. And my guess here is that this particular cell probably does not have a chromosomes inside. And when the cells actually grow without division, there's a typical signature, something went wrong with the DNA. There's a notion of a so-called SOS response, which is a system that tries to buy time. In E. coli, after doing all this experiment, The most important lesson we learned is that the E. coli will do anything to grow and to maintain constant growth. Okay? So even when the DNA gets damaged, cell continues to grow, and the cell. But what the SOS system does is that it blocks cell division while repairing the DNA damage. DNA damage is repaired, and then division is resumed. So this is typically come from the DNA damage. How long? Yes. So let me show you. Okay. So let's focus on one of the movies. Excuse me. And we'll focus on this particular channel, and especially the mother cells. Okay. Let me play the movie. There are 3 panels in this graph. The top panel is the length of the mother cells versus time. So the blue line increases cell growth, divides, and grows, divides. And every red dot is a cell division. Okay? The mid panel is the average intensity of yellow fluorescent proteins per mother cell. Okay? And you see fluctuations, but you see fluctuations around the very well-defined mean. The bottommost one is a chimograph. So this is the entire history of this particular channel. So this is intensity profile per time frame, and you basically stitch it and in time. Okay? So the question was, when the cell becomes filament, and perhaps due to DNA damage, does it ever get repaired? And then the short answer is, in general, it's very hard, but yes. As you can see here, look at this. In this particular time point, the cell continues to grow without division, becomes very big, and then it divides. And then it continues. But when it divides, it seems as if it knows how big it should be. This. And then, again, cell becomes filament again and divides. Third filamentations, boom. Fourth, and so on. Okay? So there are many statistical questions one can ask. Obvious questions we asked, for example, was that how about this interval? And what is the distribution like? Okay? And it turns out that the distribution of this distance between 2 filaments follows the power law. So what that tells us is that, well, without knowing any mechanisms, once filamentation happens, the cell remembers that it happened. It's long-term memory. Okay? Another thing is that you see here, there's very gradual climb up, ramp up, and ramp down. Okay? It doesn't become filament like that. When it becomes very big like this, still, it doesn't come down at a single division step. It actually goes through something like almost 10 generations to come back. Okay? We paid attention to this when we were doing these experiments several years ago, but we didn't understand where it comes from until we understood how the cell size is controlled. Okay? In fact, we realized this encoded everything that we need to know about the cell size control. Okay? So we'll come back to this one. If you zoom in certain part and then plot it in a semi-log scale, they're all parallel. So we know that the exponential elongation is a pretty good description for the growth of individual cells. Okay? Let's talk about this one. Okay? So the one important thing we learned is the constancy of growth rate. So x-axis is the replicative age, or the number of generations from the beginning of the experiments. And the y-axis is the elongation rate, the lambda that we talked about, of individual cells. As you can see, there's some confusing state at the beginning, but eventually they all reach steady state and then stay constant until cells suddenly die after a few hundred generations. Okay? So this is one of the most important lessons from this experiment. How about the size control? Okay. We saw that from this length versus time graph, the cells seem to know how big they should be. Okay. So how do we answer those questions? We already from the beginning, we knew the hints of size controls. So let's go back to this time series of individual cells. So we define the birth size as size at birth B, and this is division size at division D. Spans tau minutes between births and divisions. Okay. So we can plot one correlations, which is just generation time versus birth size. These graphs are normalized such that 1 is the population average. Okay? And due to stochasticity, some cells are born smaller than the population average, and some cells are born larger than the population average. If you follow individual cells like this, you measure their length at birth and measure how much time it spends to reach divisions, and you plot them, and you see this negative correlations. In other words, if the cells are born smaller than the population average, it spends more time to reach cell division. But if the cell is born larger than the population average, it spends less time to reach cell division, as if to compensate some size deviations at birth, okay? As if to correct that deviations. This is not due to the modulations of elongation rate of the cells, because as I said, E. coli will do anything to maintain constant growth rate. So if you look, as you can see here, regardless of their birth size, the elongation rate is always constant. Okay? The cell is doing something as if to reach certain desired size at division. Okay? So what's actually going on?
This is elongation rate of the finite limits.
No, this is elongation rate of the cells, yeah, individual cells. So this is a DLDT divided by L. Yeah, yeah. Any other questions? Yes. Yeah. Yeah.
What if you look at cells that are not confined?
So those experiments are more prone to experimental artifacts. Like you can grow cells on agarose pad and so on. There's the famous artifacts about aging of E. coli cells, for example. But there are groups actually who do those experiments, and they seem to reproduce essentially the same results. Okay? So nothing I'm going to say will be different if you repeat your experiments on 2 dimensions. Okay? Again, I missed the first part.
The birth size constraint, is it by an accident that the cell has been born larger, or is it like both the cells have the same size? Both the offspring, starting from a single
Both offsprings.
both offspring have the same size, which is larger than the average.
Yeah.
Or is it one of them has acquired a larger size?
So the question is, when the cell divides, and if that cell happens to be larger than the population average, does it divide in the middle or does it divide asymmetrically? The answer is, for E. coli, the division is almost always symmetric. So both offsprings will be larger than the population average. But they will all converge, as we'll tell you, converge to the population average. Yes?
This might be obvious, but will the size at division always be roughly twice the size at birth?
Hold on there. That's excellent questions. Okay? In fact, that was the beginning of everything. So that's the next slide. Satta Taheri was my former postdoc, and he was a theoretical physicist by training, did a PhD in theoretical physics, in soft condensed matter physics, etc., but probably went through a identity crisis. And he came to my lab after a short first postdoc, and he told me he wanted to switch to biology. So I was very sympathetic because I went through exactly that. Okay, trained in theoretical physics, went through a crisis, and I decided to do biology as an experimentalist. Okay. So he came to my lab because I had money for 1 year, and he started looking at the data we published. Well, that's what one would do if you're trained in theory first. Okay. And one of the things he looked at is exactly what you asked. What is the relationship between the size at division and the size at birth? Will should the division size be roughly 2 times the birth size? Well, we thought so, without thinking much, because we were very naive then. Okay, so he plotted these simple correlations. So this is the division size, and this is the birth size. Okay, so they have the same scale. So if what he said is correct, then we should have seen y equals 2 times x, right? But as you can see, this line is of the data can be very well described by a linear line, but the linear line does not go through the origin. That's y-intercept. Okay. So I overshot as an experimentalist, and I just claimed, oh, something must be wrong with the image analysis we did. Okay. So we wasted like 1 year, probably longer, looking at all different aspects of our data, the way we analyze our images, and etc. But this would persist. Okay. What's really going on? The answer came from a observation by a friend of ours. 1 second, Johan Paulsen. Before I moved my lab from Harvard to UCSD, and this is 2012. And what Johan, in our last meeting, when he saw the data from Schaechter analysis, was that the remark that what his field, people were saying in his field, in one of the models, which is about the plasmid copy number control. All E. coli cells have plasmid of some sort, which is a small pieces of DNA, often encode essential genes. Okay, so that if the cell loses the plasmid, you lose essential functions often, and then the cell can die. That's how the plasmids can survive. But there are different types of plasmids. Some plasmids exist in a low copy, meaning 1 or 2 or 3. Some plasmid exists in many copies, like hundreds of copies. Okay? So one theory proposed for copy number maintenance of low copy number plasmid is as follows, where the E. coli cell doesn't care about how many copies of plasmid it is born with. Doesn't matter, doesn't care whether it's 3 copies of 1 copy, but it only cares about adding constant number of plasmid per generation. For example, 2. You write down the equations, and after 1 line, you will see immediately that on average, this population of E. coli cells will have only 2 copies of plasmids per cell. Okay? And that changed everything. Okay? We looked at our data. We realized that instead of this line being y equals 2x, this was y equals x plus some constant. Okay? Interpretation is as follows. This is a division size. This is a newborn size. And you add some constant, just like the model proposed for plasmid copy number control. E. coli, what this tells us is E. coli doesn't care how big it was when it was born, nor does it care how big it is when it divides. It only cares about adding some constant number, adding constant size between birth and division. Okay? You had a question. How universal is this? That is one of my last slides, okay? To give you a short answer, and for virtually all bacteria we looked at, except Mycobacterium, this is true. Okay? And if you go to mammalian cells, story changes. Okay? So you can actually ask some evolutionary questions. Well, let me continue these stories a little bit, okay? Yes? Once you become filament, you enter a different regime. Okay, so if you become filament, then the division is resumed, and then it actually becomes a multiple divisions. Okay. Okay. Yeah. Any other questions for now? So we coined the adder. In the cell size control model, there are 2 major paradigms. One is the sizer, which tells you that the new round of a cell cycle is triggered when the cell reaches some critical size. Okay? For example, critical division size and cell divides. But this correlation immediately tells us the sizer cannot be true. Okay? Because as you can see, the division size depends on the birth size. So if you are born large, in fact, you also divide large. Okay? Therefore, we know that the size was not true immediately. Okay? And this correlation also tells us the timer cannot be true either. timer would tell you that the division is triggered after some fixed amount of time has elapsed from birth. Okay? As you can see here, again, the generation time depends on the birth size. Okay? We knew the data very clearly told us that neither the sizer nor the timer can describe our data. We knew there's something else. So we decided to call it adder. It's a cute name proposed by a friend, Jeff Gore at MIT. And because all E. coli tries to do is adding something. Okay? And what you're plotting here is actually the difference between the birth size and the division size. Basically, this is added mass delta, and plotted against the birth size. You can see, irrespective of the birth size, this delta, the size added, is constant. Okay? So we are very excited because we never heard about this in the literature. Okay? And so we thought that, oh, this is a really big deal. It's potentially textbook level discovery. Okay? So we had a choice. First, just write a paper, propose a new mechanism of size control in a phenomenological way. Okay? Or you be more honest and then evaluate your data very carefully. And we realized this comes from the single growth condition and single organism, not even for different strains. And somebody asked, how universal is this? Of course, if the message is very strong, you really want to evaluate the generality of that first. Okay? So we decide to do more experiments and to check these results in different growth conditions and etc. Just like we wanted to go back to 1958 and repeat those experiments. Okay? The added part is 2.7. Because again, yeah, whenever you see 1, that means we normalize by the population average. Yes. Good. At least you are looking at it very closely. Any other questions? Yes. Here. Sorry, I can't hear you very well. No. This is a division size. It's a birth size. So 2.7. This is the average. Excellent point. Wait there, okay? We'll come back to that one. So how come by adding constant mass, you can achieve size homeostasis? Okay, so that's the question. And that's one of the major points we wanted to make, and then we'll come back to that, okay? All right, here we go. So as I said, we wanted to go back to 1958 and repeat all these experiments using the tools we have, okay? And turns out that was the beginning of a nightmarish experience. And the reason is as follows. As you can see here, as you change the growth rate, growth conditions, the dimensions of the cell changes, especially the width. Okay? So if you optimize your experiments in one growth conditions, if you change your growth conditions for different sizes of cells, in fact, you have to start your experiments from scratch. Okay? So let me illustrate what I mean by that. Here, for example, everything looks happy. Let me start the movie. Here, and here. Here, everybody's happy, but eventually you'll see these E. coli cells turn into potatoes, deform, make your images totally unanalyzable. Okay? So it was very painful because even if there's a slight mismatch between the size of the cells and then your microfluidic device, the growth channels, and then you just cannot produce the data that you really need. Okay? So this was very painstaking experiment. Every experiment required the optimization from scratch. The good news is that Schaechter became an experimentalist in between. For example, this is what he was doing in the Cold Spring Harbor laboratory, okay? And he really persevered. Okay? And after several years of the effort without any results, finally things started working. As you can see here, this is the colloid cells growing using glycerol as a carbon source, sorbitol as a carbon source, glucose with the 6 amino acids, glucose with the 12 amino acids, synthetic rich medium, and this is the soytone I was talking about, 17 minutes of generation time. This is all isogenic cells, so they have exactly the same genetic sequences, okay? But look at this. Just because you change their growth conditions, what a wide dynamic range of cell size and growth rate this simple organism can exhibit. Okay? We had a lot of data after this, and of course, while we are collecting this data, we checked whether this data follow the adder principle or not, okay, gradually. And we already knew what was coming. So, like you can see here, we're plotting delta, the size added, versus the birth size in different growth conditions. Okay? So, by and large, the constants is there, although how much the cell decide to add actually really depends on the growth conditions here. Okay? Furthermore, because we have a lot of data, we can actually plot the real distributions of delta, okay, and see whether this delta is truly independent of their birth size or not. For example, if the delta is truly independent of the birth size, okay, if you bin your data for different birth size intervals, for example, here 4 groups of data, and if you plot the distribution of delta for each bin, okay, all those distributions should be independent of the birth size and collapse onto one another. And indeed, that's what we actually saw. Okay? So when you say this adder principle, that the delta is independent of the birth size, if you look at this kind of conditional distribution, the statement is much stronger than delta being just constant with respect to birth size. Okay? I'll come back to this point because we collaborate with Massimo Vergasola and then did more. Yes.
So you're not impressed by the deviation from constant line in the glycerol, for instance, the delta appears to be bigger for the small size and large,
right,
smaller for the large size.
So the question is, how seriously should we take about this apparent deviations, okay, from the perfect horizontal line? And then I should say 2 things. The first, if you look at the distributions, these deviations come from really very small fraction of the data, actually. Okay? So from statistical point of view, we didn't take those deviations too seriously. But as you will see, all that matters turns out to be whether this follows the linear line or not. Okay? Whether delta versus newborn size can be described by a linear line or not. Because as soon as that line, this follows the linear line, then all the major claims we make essentially stay the same. So we don't too much worry about these deviations at the tips, for instance.
So the deviations for, except for the last high growth rate,
yeah,
where the deviations seem to be in slightly opposite direction. Here the deviations, all the deviations are sort of
slightly negative, for example.
right.
Yeah, in this case, yes. So here, for example, there's slight deviations, etc., etc.
Yes. So all of them, and the last one, which is the new growth medium that you have, right? The very fast one. So then it's slightly different, right? Is there something different about the new growth medium?
So the short answer is that, yeah, you can model whatever we want. In fact, we even had a model that explains how this transition from, say, negative 0.1 to positive 0.05, for example. Okay, we can fine-tune those models, but in the end, we decide not to do that. Okay. For the reasons I explained. Yes?
But these deviations are interesting. I think they would predict a faster convergence towards the average size than if it was totally flat. Do you see a faster convergence?
Not in this, we didn't particularly look for it, but the negative slope indeed imply the faster convergence because it should behave like a sizer. maybe I should actually wait, but let's wait that discussion a little bit later, because we need to first describe why this perfect adder ensures the size convergence first, and then we can talk about the implications of different slopes. Okay? Any other questions? How general is it? It's fairly general, because we also looked at the B. subtilis. Okay? B. subtilis is a gram-positive organism, which is 1 billion years divergent from E. coli. The distance between E. coli and B. subtilis is further than between me and elephant, for example. Okay, so it's a very, very divergent. As you can see here, in 4 different growth conditions, the adder is more or less ensured. Okay? How about the size mutants? Okay? If you look at the cell cycle, history of a cell cycle study and development, and the key was to studying mutants. Okay. So we looked at the size mutants as well. For example, this particular mutant is FTSA, has to do with the defect in cell division, for instance. But again, although how much the cell has in terms of size between birth and division differ numerically, but the constancy is still there. This mutant is about the defect in cell wall synthesis, delta PGM, and again, the constancy is there. So by now, we've tried many different mutants and played with the different strains and etc. We just cannot break this constancy. Okay. Except one case, which I'm not going to talk about. Why should we care about the perfect adder? Because if you take it seriously, it ensures the size convergence. Also, just like the one gentleman over there asked at the beginning. Imagine E. coli cell that's born much larger than the population average. Okay, the cell adds constant delta and divides in the middle. So in the next generation, the daughter cell becomes slightly smaller than previous. And then, but it adds, again, the same delta and divides in the middle. Next generation, it becomes even smaller. You can see this will continue until this daughter cell itself becomes exactly the same as delta itself. Okay? Then it will stay there. How about the cells that are born much smaller than the population average? It adds constant delta and divides in the middle. So next generation, this time, the daughter cell becomes slightly larger than the previous mother. And this will continue until the size of the daughter becomes exactly the same as delta itself. Okay? So no matter what your newborn size is, the cell is guaranteed to converge to the population average. Okay? Of course, the real picture becomes a little more complicated because you have to add the stochasticity and so on. Okay? But this general principle stays true. Okay? Yes? Exactly. Yeah. Delta is the average newborn size of the populations, okay? Which has very important implications. Any other questions? Yes? Correct. Exactly.
And there is that temporally correlated. I mean, that's, I'm trying to ask whether that's how you end up with smaller and bigger cells in the first place.
Yes. So you remember, I plotted the delta against the birth size, but I also showed you the distribution of delta itself, which was not a delta function. Okay? So delta itself has a stochasticity. Okay? Sometimes you add slightly larger than 2.7, sometimes slightly smaller than 2.7. Okay?
So, if at a given time, a given cell is adding let's say more, then does it continue to add more and then?
No. No.
There's no temporal.
No. So delta is independent from each other. Okay? If you take the computer correlations, it decreases exponentially.
than its population average or less than its population average, because the division happens very precisely.
Wait there. We'll come back to that point. Okay. Yeah. We'll come back to all the distributions and etc. etc. Okay. Yeah.
So that means that when you are going to different mediums, the deltas are changing.
Yeah.
And then this should also fall in the exponential line of the growth block.
Excellent. Excellent. Where you already, you already spoiled it. That is one of the spoilers. But what he is pointing out is that, also, there's another student mentioned it, that there, delta is the population average of a newborn size. Okay? So after more than 50 years since the Schaechter, the growth law, we know where the y-axis comes from. Okay? And what the cell really tries to control is really the delta itself. This is nothing but log of delta. Okay? So after more than a half century later, we've made half step forward. We know where y-axis comes from. Okay? But we still don't understand why they follow the exponential line. We have some idea. Okay. All right. Okay. Before people give me more spoilers, I'll go to the next slide. Okay. So how general is it? Well, it is a comparison between the data and the model. There's no adjusting parameters. This is E. coli and this is B. subtilis. Okay. So pretty happy. The size convergence. Okay. But because we had a lot of data, we realized that we can extract a lot more information than what we just talked about. So 1 year after I moved to UCSD, Massimo Vergasola also moved his lab from Pasteur Institute in Paris to San Diego. And then we immediately started collaborations, and we divided our task between experiment and theory. So Massimo and his postdoc Serena Brade looked at, started from our data. So our starting point is this conditional distributions and their collapse, for example. Okay? But now look at the shape. They look kind of Gaussian. I mean, these days, you just plot the distributions, everything looks kind of either Gaussian or log normal. Okay? But it doesn't matter whatever that is. What is important is that, as you can see, they all have sort of a similar shapes. Okay? So this similar shape was something we already knew from the beginning. And then when I mentioned this to Massimo, and he had some critical insight from statistical mechanics. So for example, as I said, all these lines, if you rescale the x-axis by the respective mean, then they all collapse into one another. Okay? The question is, where does it come from? Okay? And the second question is, what is the implications of this kind of universality or the collapse of all these distributions? Okay? And this feature, in fact, is true not only for E. coli, but B. subtilis. B. subtilis, if you look at it, it has a more longer tail. It looks, the delta distribution is more asymmetric. But if you look at the size mutants, for example, even size mutants follow this collapse. Okay? So this is a very general feature of this kind of size control. Now, if someone says something like, oh, the cell divides very precisely in the middle. So I have some quiz for you. Let's look at this time series. Okay? And this time series implies 6 parameters. The cell is born at a certain size, and it spans tau minutes, and then it divides at size SD. Okay? Then the cell has to divide. A cell has to decide where to divide. Okay? And then there's a delta, which is a size added. And also there's the elongation rate, which is a DLDT divided by L. Okay? So some of them are related, but there are total 6 parameters, immediate parameters we can think of. And we have 6 histograms, sets of histograms. For each histogram, as you can see, has multiple lines because they all these histograms come from 7 different growth conditions. And if you rescale them by their respective mean, they look more or less collapse onto one another. The quality of collapse is pretty good for certain parameters and the less good for some other parameters. Now, I deliberately removed all the labels on the x-axis, okay? But as you can see, I plotted them in the order of increasing CV, okay? The distributions. Which parameter do you think shows the narrowest distributions? Okay. You have 1 out of 6 chances of winning. Any volunteer? So this guy. Okay, good, very good. How about the next one? Which will show the second narrowest distribution. And again, edit size. Who says that? Why? Yeah, because it's a control parameter that I try to convince you the cells really care about. Okay, so it's natural to think, oh, the distribution of that control parameter must be narrow. That's what he thought. Turns out that it is elongation rate, followed by the division size distributions, newborn size distributions, generation time, and delta itself shows the widest distributions. Any idea why it may be so? But now, in the early days of systems biology, this width is exactly what people called the noise, right? So delta is the noisiest one, although I've been really trying to convince you this is the parameter the cell really cares about, okay? Think about it. I'm happy to chat about it, okay? So what's next? So what Massimo and Serena decide to do is that, how many of these, what are these parameters out of the 6 parameters are really fundamental parameters that really cells care, okay? And how many of them are really independent? Well, the way you go about that is very simple, in fact. All you do is that you just plot 2-point correlations, okay, 2-parameter correlations for all pairs of correlations. So it looks complicated, but actually it's not. So let's just look at this particular case. Here, the y-axis is lambda, the growth rate, and we're plotting that against, it's cut, newborn size, delta, division length, etc., etc. And as you can see, this is a flat, 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 flat. So this is not correlated with any other parameters. So you can see that this is a parameter that the cells control independently of all other parameters. So it's safe to assume, well, this is one of the very basic parameters that cells control. And then you basically iterate this logic, and then we settled with the 2 parameters. One is the elongation rate, and the other is the delta itself. Okay? The next, what you do is that you check the consistency. This time, you start with the basic parameters of our choice. One is elongation rate, and then edit size. And then you basically write down the mathematical descriptions of growth and division processes, and taking these 2 as the input parameter of your modeling. Okay? Then if our claim is truly correct, that these 2 parameters are 2 independently controlled parameters, and then are sufficient to describe all our statistics, then by taking these 2 as input, we should be able to reproduce all other parameters and their distributions. And indeed, that was the case. Okay? So this is the simulations, comparison with the simulations and the data. And they also did even further, and then understood that if we take this argument seriously, can actually show that how the CVs of these 4 distributions must be related with one another. And these parameters are the division size, newborn size, generation time, and edit size. In other words, if you know the distribution and the width of this particular parameter, you can actually explain or predict the width of all other parameters. Furthermore, if this guy collapses, everything else has to collapse. So the other parameters also should inherit these scaling properties of delta itself, okay? As you can see here. Right? So we are very happy. So we didn't write this explicitly, But our current view is that during evolutions, cells somehow have evolved to learn to control two different types of parameters. One is the growth control, which is about kinetics, it's about time. The other one is a size control, which is a delta. And these two talk to each other through metabolism and physiology. And everything else, such as generation time or newborn size, and etc., are slave to these two basic parameters, control those two parameters. Okay? So this is our current view. To what extent is ether general? Somebody asked, okay, how general is this? It's fairly general. And when we're sitting on this for more than 3 years because Sata was generating, trying to make his experiments work. But by the time we're trying to publish this, and it happens in biology quite often these days, somehow we hear that, another group is doing the same thing, another group is about to publish this, and etc. So I was really forced to think, look at all the published data, and to see whether this ether, what he called ether, is also valid beyond E. coli and B. subtilis. So let me show you some data. This is from the East Cerevisiae. This is from Fred Cross lab at Rockefeller. And I'm sorry, this is Colobacter crescentus. This is data from Novosher and then Arundino's lab in Chicago. And if you look at their data carefully, there's almost constancy here. Okay? And this is East data from Fred Cross lab. And the East, depending on how many generations the East cell is old from previous generations, can either follow the ether-like, it can show ether-like behavior, we can show slightly negative slope. Okay? And if you look at the FANTEST 1977 data for Fusionist, excuse me, this is a very strongly negative. Okay? So what's the meaning of all this? And as Professor Pipel said, the difference the different slopes make is about the speed of convergence. Okay? If you have a very negatively, strongly negatively slope, in fact, if it's slope -1, there is a perfect sizer. Okay? You can actually show immediately that after just one generation, you'll reach the target size. Okay? Whereas if the slope is just 0, and the convergence is a geometric series. Okay, so it's exponential. You have to wait. You have to divide several times to really reach the final target value. Whereas if the slope is +1, perfect, there is a perfect timer, and there's no stability there. And the whole size dynamics becomes like a random walk, and the size distribution just diverges. Okay. Actually, I have one slide that I'll come back to that one. And this is data from Christian Jacobs Wagner's lab at Yale. And look at this. This is a Colobacter, and this is a Colobacter. And this time, they say even Colobacter crescentus follows the perfect adder, slope 0. Okay. No. Thanks God. Correct.
What if the partitioning between the cells is unequal? I mean, for example, in this Colobacter crescentus thing, you have a stalked cell, and I mean, there are two sort of cells that the cell divides to. So, I mean, and if you have a timer then, and you have this unequal partitioning, would that also lead to like instability in the distribution? I mean, would that have size homeostasis is the question.
Yes. So your question is?
The question is that you said that the timer leads to unstable distributions. They would run off. I mean, they wouldn't stabilize. All I'm saying is you add partitioning. I mean, if the partition is unequal.
No, it's not gonna. It's gonna save Colobacter any time over there. Okay. And simple reason is that if you think about why timer cannot work. By the way, timer can work if the cell grows linearly, for instance. Okay. But the timer doesn't work if cells grow exponentially. Okay? Because what timer would do is that whatever the newborn size is simply double. Okay? So if you're slightly bigger than your target size, okay, and this cell size will become, it will increase exponentially. Okay? But by introducing, so in Caulobacter's case, cells divide slightly asymmetrically. Okay, they grow exponentially. Do I have a slide of that? Shoot. What time am I supposed to finish? Give me 2 minutes. Okay, yeah. Okay, the short answer is that the Caulobacter, despite they divide asymmetrically, they converge very nicely separately. Okay, I'm essentially finished. So this is basically summary of all the data, and most of the bacteria we looked, excuse me, we looked at follow this perfect adder. There's some deviations of this microbacteria, but it doesn't grow exponentially. Okay, so which is okay. And this is at least one group shows that the Caulobacter crescentus may not be perfect adder. So this has to be resolved by these groups eventually. Okay? And if you look at the mammalian cells, and it's not here, mammalian cells or most yeast cells, they behave like a sizer. So convergence should be faster, just like I say here. Okay? The sizer would converge to the target site after one division, and then this, the adders will follow the geometric series. Okay? So to summarize, I talked about it. This was the original Schaechter line, 1958. And as I said, after more than a half centuries, we've made a really half step forward. We know where the y-axis comes from. It is really about the control of the delta. Okay? The question is, how does the delta really change in a different physiological state? Second one, question I asked at the beginning was that is there anything we can predict at the single cell level? And the answer is yes. So this is one of the typical movies that we have. And I'm going to show you. And we start the movie. And this line is a predictions based on the what delta we have. Okay? And this is the average line. Now look at this. Growth and divides, next generations. Okay? Following this geometric series. Of course, because of stochasticity, there's some deviations, but deviations around the very well predicted the line. Okay. So I guess this is it. Okay. So this is where we are in terms of understanding of size control. And there are many, many open questions. Okay. And I think from my point of view, all the open questions about the coordinations. Okay. We know a lot about molecules. We know a lot about the control of individual cell cycles, such as replication control, cell wall synthesis, growth, et cetera. But I think the real questions are, how are all these different stages or aspects of growth are coordinated? You perturb replications, how does that affect the rest of the physiology? If you perturb cell wall synthesis, for example, or if you perturb the shape of the cells, how would that affect everything else? Okay? This coordination issue is a virtually unexplored virgin field. Okay? And we have a lot of data, like many other groups. And this is also just like Professor Peter said, where even theorists, and not even, actually theorists, can really make some contributions. Okay? So if you have any ideas, you want to talk to me, you're welcome to send me an email or talk to me. Okay? Thank you very much for your attention in the last 3 lectures. So thank you.
How does it relate to your added hypothesis? How does two relate?
So the question here is that I talked about the plasmid copy number control, and what does it have anything to do with the size control? And the short answer is they have nothing to do with each other. And the connection is the formal analogy, okay, at the mathematical level. So that is the only thing.
This is with respect to the Schaechter law.
Yeah.
You have the value of delta for different growth conditions in which the growth rates are different. And so you also have, so therefore you can also check the Schaechter law, right? You didn't present your data about the Schaechter law.
Our data follows exactly the Schaechter law, implying C plus D equals 67 minutes.
Implying?
C plus D is 67 minutes for the strain we used.
So Just there's a question there that does it, so there is a exponential growth of size with respect to growth rate. Does the data permit a subleading effect which is not exponential? Let's say something which is modulated by some prefactor in front. Does the data, is it exactly fitting an exponential, or is the data
Growth law, you mean?
Yes.
So as always in biology, there are always exceptions. And in this case, this is not really pure exponential. And if you really look at the data, I believe it plateaus. So as the growth rate really decreases below a certain point, the typical breakpoint is like 60 minutes of generation time. If you go below that, everything breaks down. Having said the Cooper model, C is not constant anymore. The C period increases very, very abruptly below the generation time 60 minutes, and even that also deviates. Okay?
So for slow growth rates, it deviates, but for higher growth rates, it agrees with this exponentially.
As I can see from our data and other people's data, this is true for fast growth conditions. Yes?
Value delta varies depending on the growth conditions, right? So how will it be when the bacterium is grown in a particular growth condition and then transferred to a new growth condition? So how will the delta change?
Excellent. Okay, that's exactly the kind of things, experiments we're trying to do right now. So if you did the shift up experiment at the single cell level, okay, what will happen? Here, I think the deeper question you are really asking there is that during what stage of cell cycle does the cell commit to a particular value of delta? Okay, so that is the real question. And we don't know the answers, although we've done the analysis. And then right now, there are two camps. The first camp says that delta is cell commits to new delta upon division, and it tries to add a constant delta from division to division. The other camp says that the delta is added from initiation of replication to next round of the initiation of replication. As far as our data is concerned, we can both camps produce consistent predictions with the data we have. And to really differentiate to these two camps, we really do need to do the shift up experiment, among others. Then we'll see, as I said, shift up is like a prism, then we'll be able to tell. And we're waiting for the data, but it's very challenging experiments. Lunch? Okay.
Okay.
Thank you.
Thank you very much.